I'll tell you today about a couple of projects on how cells polarize, self-polarized, and initiate motility. And so just a very introductory slide on cell migration, which is a fundamental process in cell biology. There are many examples of it. Cells migrate when there are cancer cells that are metastasizing and trying to crawl through tissues to blood vessels. So this is, of course, sinister example. A good example is wound healing. Cells move into the gap, filling the wound and creating scar tissue. And in development, pretty much all cells at some point are moving from place to place. And understanding cell migration in physiological systems is very hard, because cells move normally through three-dimensional space with very difficult geometry and topology. And so for decades, people were studying how cells migrate simply on flat surfaces. So this is a view from the side. And that's what I'll be talking about. And unfortunately, today, I will not have time to talk about 3D migration at all. Just believe that some lessons from 2D actually useful in 3D. However, there are different opinions also. So this is basically one of the cell biological dogmas from any textbook, which explains in a very schematic way how cells migrate. So they have to make protrusion, creating the new leading edge. They have to create contraction inside, and they have to create a certain type of adhesion sticking firmly to the substrate at the front and only weakly at the rear. And then this contraction inside pulls the rear forward, finishing this cycle of locomotion. So to kind of loosen up the room a little bit, let me show you a movie which is one of the most famous movies on the web. So you probably saw it. If you didn't, this is a movie of neutrophil, a cell of our immune system, chasing a bacterium. And all around it is red blood cells. So note that this neutrophil, it's not swimming, it's crawling on the inner surface of a blood vessel. And so in the end, it gets bacterium with satisfying sound. And so, as I said, this is one of the most famous movies on the web. What is more fun is than watching this movie is to read the comments on the web, which is really amazing. It kind of well, after you read them, you kind of stop being surprising that so many people support Donald Trump. But anyway. But anyway, it basically shows that studying cell migration is really important. So, as usual, it is very important to focus on very specific organism, very specific model system. And for decades, we were very interested in fish epithelial keratocytes, which are very similar to our fibroblasts. Those are cells from connective tissue of fish skin. So let me show you again the movie of those cells. The movie of those cells migrating. So here you see the cell from above. And basically why people studied them for so long is that unlike many other cells that move in principle in the same way, but in fact the shape changes a little bit, changes a lot. The velocity fluctuates. Everything is very volatile. So as you saw, keratocyte cell, it moves very steadily with pretty much constant shape, with constant speed for hours. So it's a great system to study. So the main thing to understand about this motile system is the following. If you compare the view of the motile cell from above and from the side, so what you see here at the rear is this huge three-dimensional blob which pretty much contains all the cell organelles. There's nucleus, there's ER, Golgi, everything. Everything is here. And this ultra-thin but very broad and long appendage is the lamellipod. And this is the motile appendage of the cell. This is the engine of the cell. There are pretty much no organelles inside. So latest research says that there are no even mitochondria. So it's basically just cytoskeleton wrapped in membrane. And there's a very simple experiment showing that lamellipodium is the engine and the cell body is basically a passive mechanical cargo. There are many ways to cut off or make the cell spit parts of the lamellipodium, and those so-called cytoplasmic fragments, they move pretty much like a mother cell with the same shape, with the same speed for hours until they run off ATP, and then they of course stop. While the cell body alone cannot do anything. So the point is to understand the lamellipodium. Before I get to the model that we use and to actually the problem, the important thing, basic thing about the lamellipodium is the following. It is filled with actin-myosin network. And this network is self-organizing into the so-called branching dendritic network. So what's happening is near the leading edge of the cell, actin filaments, which are long polymers that are polar. They have plus end, which is growing rapidly, and minus end, which is disassembling. And then monomers, which are disassembling from the minus ends, are diffusing, engaged in tens of chemical reactions, and assemble again onto the plus end. And so what's happening is each existing filament which pushes the leading edge forward, it grows for a while, then gets capped by capping protein, and then a new filament appearing from the side of existing filament, and so this networks kind of propagate this way. There are a huge body of research on this system in the lamellipodium, and it's one of the examples similar to bacterial chemotaxis, for example, or cell cycle, where we know a lot. We know not only all the molecular players, but the concentrations of them, the rates of reaction, a lot of stuff. The main point here is that closer to the leading edge, this actin network is mechanically very firm and solid. But as you go away from the leading edge toward the rear, because there's disassembly everywhere and assembly mostly at the leading edge, the network is getting less and less dense and more mechanically weak. And there are also myosin everywhere in the cell, which is this protein. You of course know that actin and myosin are two main players in the muscle, so myosin tries to collapse actin networks. And so myosin is mostly at the rear, creating contraction which pulls forward the rear. So that's sort of the qualitative understanding, and what you need physical mathematical model for is to see if this understanding makes sense from physical point of view, and see if this model predicts something interesting. So what I'll do today is I'll show you a couple of things. One is how this model explains steady movement, but another I wanted to explain to you how the cell initiates motility, and it's really beautiful process kind of irresistible for a physicist. So the cell, when it starts, it sits on the surface in a symmetric disk-like shape, and then completely by itself, without any extracellular cue, it loses symmetry and start moving. So the cell body in the middle, it looks like a fried egg in the beginning, mound of the cell body and ring of lamellipodium around, and then it self-polarizes and starts moving. And so I'll show you how a very simple model explains exactly this process. So here is the model. It consists simply of two partial differential equations. This is the minimal model. We have the whole hierarchy of those models. There are some of them have more equations, more parameters, and more sophisticated. But this minimal model explains the essence of this self-polarization. And so it's two equations. The first equation is balance of forces in the lamellipodium. And so if you take any small chunk of actin-myosin network, there are three main forces. One is passive forces due to deformations of actin network. So if you try to shear it, it resists. If you try to compress it, it resists. And lots of biophysical experiments showed that, roughly speaking, on the time scales that interest us, which is tens of seconds and minutes, actin network behaves like a very viscous fluid, like honey or molasses. So on fast time scales of second or faster, it is elastic, but on slow time scale, which is of interest here, it's viscous. And of course, those viscous forces sometimes are described by complicated mathematical expressions, but this is kind of the minimal description of this viscous stress. We have models when it's viscoelastic and much more sophisticated than that. Then the second force is from myosin contraction. So M is myosin density, and basically the assumption of the model is that stress generated by myosin is proportional to myosin density. And so if, for example, myosin density is constant, there will be a lot of stress but no force, because myosin will contract here and here with the same strength, and in the middle nothing is going to happen. But if there's more myosin here than here, then contraction here is stronger, and there will be a force which is a gradient of the stress, which is a gradient of myosin density, and K is proportionality coefficient. And finally, there's a force of effective viscous drag when actin network starts moving relative to the surface on which the cell is sitting, because there's lots of molecular adhesions between the actin network and the surface. And so this force is modeled by the usual expression. So the force is proportional to local rate of movement of actin in the lab coordinate system. And this is effective viscous drag coefficient. So one note is that this force of adhesion force, which is often called traction stress, can be easily measured these days. The cell is simply placed on a flexible substrate with fluorescent beads embedded in it. And from deformation of the substrate, you can measure this force. Inside the cell, those forces are impossible to measure, and there's just no idea how to do that. But this one can be measured. Right, so this is force balance. And then the second equation, to solve this equation, we need to know where myosin is. And so equation for myosin density, this is, you can call it mass conservation law for myosin or transport equation for myosin. The important term in this equation is the term describing a drift of myosin with actin. So roughly speaking, myosin simply drifts together with actin network on which it's sitting. But there's also myosin molecules are detaching from actin, floating in the cytoplasm a little, attaching at another place, and this is effectively myosin diffusion. However, this term is very small. Estimates show that it's very small, and it's numerically good because it's smoothing out those equations, but in principle it's not important. And so the advantage of such model, it's very simple, very intuitive, and it has only 3 parameters. So diffusion coefficient, as I mentioned, very small and nothing depends on it. It's viscosity of actin network, strength of myosin, and viscous drag between the actin and the surface. And from many experiments, we know the order of magnitudes, magnitude of these parameters. So another thing important to realize about this model, there's this feedback loop which the model qualitatively predicts. This loop is easy to understand this way. If you have aggregate of myosin molecules in this place, right, they create, myosin creates the flow which goes up the gradient of density. So there will be flow bringing more myosin in this gradient. And the more myosin there is, the higher the flow that brings even more myosin. So it's easy to understand that if, for example, viscosity and diffusion are small enough, the prediction would be that all the myosin will collapse into one point, creating the flow which leads into this point. So in this sense, the model is mathematically rather similar to famous Keller-Segal model of chemotactic aggregation of slime mold, which is one of the great paradigms in mathematical biology. So the first test of this model is as follows. We can, of course, readily solve the system of partial differential equations on the shape resembling stationary and moving cell and see what the model predicts. So in the stationary state, when the cell is disc-like, so we assume that adhesion is constant everywhere, and then the model predicts, as I said, myosin condenses into the middle, and then it creates centripetal flow, bringing even more myosin into the middle, so it's very intuitive. If we take the shape resembling the moving lamellipodial shape, and one thing is, if we want to solve everything in the coordinate system moving together with the cell, we have to introduce the flow of effective flow of actin from the front to the rear. So if we do that, then the prediction is that myosin will all accumulate at the rear, right? Because actin network just sweeps the myosin to the rear, and that's where the most of the flow will be, because you need high concentration of myosin and high gradient of it to create this flow. And so experiments indeed confirm that all those are the cases. So if you take stationary cell, you have centripetal flow. It's of course not completely uniform because nothing in biology is constant and uniform. Myosin, so the complication is because of the cell body. Myosin is bunching up around the cell body. It's probably under the cell body also, but we don't see it in the experiment. And when the cell is moving, the flow is high at the rear as predicted, and myosin also is swept to the rear. So the real test of the model, however, is would it actually predict the shape of the cell, which is observed, right? So it's much easier to figure out how things move on those shapes, but how those shapes are evolving. So to do that, one have to solve more sophisticated mathematical problem. We have to add free boundary. We have to allow the boundary of the cell to deform. And so the rule of how you deform the boundary is also pretty much clear from the experimental situation. So as I mentioned in the introduction, actin is actually growing at the boundary, right? So without myosin, that will be all that ever happens. So we introduce the following process. Each point of the boundary expands in the locally normal direction with constant rate. There was actually an experiment in '93, famous experiment showing that indeed the boundary expands in a locally normal direction. Experimentally, it's known that this rate of growth is not in fact uniform around the edge, but that's kind of only complicates the model and is not really necessary for the minimal mechanism of polarization. So what without this growth, on the other hand, another thing is this rate of growth, of course, is slowing down when the area of the cell starts to increase too much because there's membrane tension. Actin has to push on the membrane. If there was no myosin, the prediction would be that the cell is completely disc-like and it expands slower and slower and slower. With what myosin does, as you saw, it predicts the inward flow of actin. So actin tries to basically collapse. And so what it does to the boundary, it moves each point of the boundary inside with the rate predicted by this model that I already showed. And so the net rate for the boundary expansion or contraction is simply geometric sum of those two velocities. So that's how we move the boundary. And then we use level set method to actually do the simulations for those solving this partial differential equations on moving boundary and seeing what it predicts. Yeah.
Are those two equations on the left also boundary conditions?
So, right, so those are boundary conditions, right? These are equations that I already showed. And and boundary conditions are very simple. There's no flux for myosin and zero stress for force balance equation. Right. So when we solved this model, the result was the following. The cell actually, so we solved the model with initial condition such that myosin is slightly biased to one side of the cell. And the slight initial bias creates the initial flow, right? And then see what happens at this boundary. This boundary starts the net movement in this direction because this inward flow overcomes the expansion of the boundary due to actin growth. Here the growth of the boundary is faster than inward motion. So this boundary starts moving this way, this boundary starts moving that way. As a result, some gradient of myosin density persists. Myosin keeps being swept to the rear. And so this feedback, which is in the model, is enough to polarize the cell by itself. You start moving the cell, myosin is swept to the rear, that's where it pulls in the rear. At the opposite side, front outruns it, here you are. However, clearly the predicted shape of the cell is very far from what is observed in the experiment, and also the predicted speed of movement of the cell is 2 orders of magnitude slower than what we observe in the experiment. I remind you that we actually know the parameters of the model. So this feedback is really not enough. So the hint of what we're missing comes from the following observation. So far in the model, we assumed that adhesion is constant. Strength of adhesion is constant. But the strength of adhesion can be estimated if you actually divide the force of adhesion, force of drag due to adhesion, by the local flow of actin. And as I mentioned, the force can be easily measured these days, and flow can be also measured from fluorescence spectral microscopy. And if you look at the forces and the flows in the stationary cell, you'd see that the forces are rather high and the flows are rather slow. While in moving cells, for example, in this region, forces are very low and the flows are high. So obviously, the adhesion is not constant. Okay. And so when we plotted the traction force as a function of the speed of actin flow, the constant adhesion would be just straight line. But in fact, there's this biphasic dependence of the force on the flow. So the force is growing linearly with the flow as it should first, but at high flow it just collapses. And this is not a completely new thing. Other groups were measuring the same type of dependence for other cells. So what this phenomenon describes is the so-called stick-slip adhesion. And it's a very simple mechanical phenomenon which actually exists in our microscopic world as well, for example, in dry friction. So if you move something with slow rate, adhesion is high. When you started to move this object fast, the strength of adhesion drops simply because fast movement breaks molecular bonds of adhesion. I have to mention that in cell biology, the opposite phenomenon is also known and also famous. When you pull some types of adhesions and they become stronger when you pull on them. Okay? But so apparently in keratocytes, it's not this more fancy type of adhesions, but stick-slip adhesions. And so mathematically, this can be described really easily. You simply take this data and divide force by speed and see how this adhesion coefficient depends on velocity, and it just depends like that, threshold type. If velocity is lower than critical, high adhesion, higher than critical, low adhesion. Qualitatively, it adds this negative feedback to the positive feedback that we already have. And so when we added this term, then and added some initial fluctuations to the cell to destabilize it in the beginning, then the cell spontaneously loses symmetry and assumes quite realistic shape, starts moving fast, and also has this pattern of traction forces very similar to the experimental one. And the explanation, of course, is very simple. As far as initial adhesion is low enough at one side, or myosin is high enough there, the higher than usual flow breaks adhesions there. This end retracts, allowing the other sides to expand. And this is irreversible. Then the cell is locked in this position because this rear is switched into the sleeping state, and this remains sticking state. I have to mention that in the experiment, that's actually exactly the result. It's hard to keep the state stationary and symmetric. As soon as you kick it into model state, it moves without stopping. It's very hard to stop it. So we do have bistability, but the moving state is more stable in a way than the stationary state.
What about the speed? You said in this simple model, the speed was by 2 orders.
Right, and this one is of the right order of magnitude, yeah. And so there are many tests of this model. I don't have time to show you all of them. Well, just one of them is, so this is, for example, what happens in the simulations when you start from initial condition, when you lower adhesions at one side. Then the cell retracts from that side, starts moving in the opposite direction, and that's exactly the result of the experiment. When you blow from micropipette the solute which weakens the adhesion at one side of the cell retracts from this side and starts moving in opposite direction. And of course controls to show that it's not just mechanical effect of the flow. So that's pretty much the first part of my talk. And let me skip that. So that's the conclusion, that there are two feedbacks which are absolutely crucial for this self-polarization. So another thing to mention about the self-polarization mechanism that is purely mechanical. There are lots of known mechanisms when you have, for example, Turing-like mechanism, when you have signaling molecules creating polarized pattern, and this polarized pattern then tells mechanical system what to do. So we have enough evidence from the experiment after comparison with the model, to say that keratocytes can polarize completely mechanically without any of this chemical system upstream. We cannot prove it definitively, of course, but there were experiments with inhibiting some key signaling molecules, and keratocyte still polarizes fine without those signaling parts. So I thought in the second part of the talk, I'll do two things. I have experimental part of my lab where we do experiments on galvanotaxis, on how cells move in electric field. And also, this won't work, never mind. And also I'll show you another model of how different type of cells break symmetry. So on galvanotaxis, so this very same keratocytes that you saw, they are not known to be chemotactic. They're very peculiar in this respect. It doesn't mean that they are not chemotactic. In fact, it's simply that nobody yet figured out what's the chemical they would be attracted to. But they are well known to be galvanotactic, and so they prefer to move to the cathode. Unfortunately, this movie doesn't work on the computer, but just take my word for it. People actually don't know much about the galvanotaxis, though this topic is becoming more popular. It's actually not a fluke. Pretty much all the cells sense electric field and like to move either to the cathode or to the anode. One of the functions of this process is that in wounds there are normally strong electric fields with the cathode inside the wound. And cell sensing the electric field and moving to the cathode accelerates wound healing sometime order of magnitude. The reason for this electric field is there are strong ion fluxes across our epithelial layer. And so if you break epithelial layer, the hole is just like a shortcut in electric circuit. Ions start to flow through the shortcut, creating this electric field. So there are many experiments showing that if you disrupt electrical field in wound, a wound heals a few folds slower, sometimes order of magnitude slower. There are actually even commercial devices that are used in some hospitals in really bad bone breaks. You put this device on, it creates electric field. There's actually a debate. Many people think that it's a bogus, and I'm of the same opinion. It's complete fake. So galvanotaxis is real, but those devices have no purpose at all. But you know how American medicine works. I'll just mention one interesting thing about how electric field affects this polarization of the keratocytes. So you saw that keratocyte can polarize by themselves. So what we did was we put the stationary keratocytes in electric field, and they also polarized and started moving. So the difference is that spontaneously it takes tens of minutes for the cell to start moving. And I didn't mention, so one of the things that the model predicts is this few tens of minutes which are required for self-polarization. So the big difference with electric field is it takes only a few minutes for the cell to polarize and start moving. So these are heat maps of protrusion and retraction around the boundary of the stationary and then polarizing cell. Warm color is protrusion, cold color is retraction. So in the cell without electric field, first there's this chaotic pattern of protrusion and retraction, and then retraction starts at the future rear and the cell starts moving. What's happening in electric field is that immediately all the retraction is polarized to anodal side of the cell and protrusion is polarized to the cathodal side. And so the hypothesis would be that it's exactly the same mechanism of polarization. It's simply that electric field somehow triggers signal transduction inside the cell, which polarizes protrusion and retraction to opposite side of the cell. However, there's very simple experiment. If you put the cell in electric field, it polarizes and starts moving, but then you switch off the electric field and cell depolarizes immediately. So it's very unlike the cell which polarizes spontaneously. So if it was polarized before electric field and you switch off the electric field, it keeps moving. As I mentioned, self-polarized cells, it's very hard to stop. They are in motile state. And so it suggests that there's a very different biochemical pathway of polarization of cells in electric field. And indeed, when we started to look at cytoskeletal markers, then actin and myosin are distributed in a very similar way in stationary and motile cells with and without electric field. But adhesive proteins, there are many of them. One of the most important ones is vinculin. In stationary cells is distributed the same, but in motile cell it's distributed differently. Cell which polarizes without electric field, all these adhesions are swept to the rear. They're relatively weak adhesions at the front. But in the cell polarized in electric field, there's this bright rim of adhesive proteins at the front. And so the hypothesis we're working now on testing is what's really the mechanism, but this hypothesis is that electric field just somehow induces weakening of adhesions here, but it doesn't put them into the stick-slip mode. As soon as electric field is switched off, adhesions restore at the rear and the cell stops moving. And so just one of the indications how complicated things are. It seems that this induced polarization has different mechanism molecularly than self-polarization. Let me skip that. And so the last thing I wanted to tell you is how cells spontaneously polarized, how different cells spontaneously polarized. And so sometime after talks like that, people ask me, how general is your model? Is it applicable to other cells? And unfortunately, however much I want to say that they're general and applicable to everything, the answer is the opposite. So all these mechanisms are very specific. And the second project shows that some cell, which is not dissimilar to keratocyte, it's actually very similar, uses very different mechanism for self-polarization. It also involves actin and myosin, but in a very different way. So let me tell you about that. And by the way, all of this is published. So the first part of the talk was in PNS last year, and this project is in Nature Cell Biology also last year. So this time we worked with one of these rat liver cells. It's also epithelial cell. So when it's on the surface, it's also disc-like. Like keratocyte. Nucleus is at the center. The big difference is that myosin, which in keratocyte you saw in one of the figures, it's bunching up around the cell body. Myosin is at the very periphery of the cell, creating this bundle. And in fact, when we looked closely at this bundle, it's actin-myosin bundle, very similar to contractile ring, which is used to in cell division to split the cells in two. It's not completely clear why, and from laser ablation experiments, we know that this ring is under tension. So we don't completely understand why it's not collapsing into the center of the cell, most likely because there are very strong adhesions along this ring that doesn't let it collapse. And so another thing is that when you look closely, there's this actin-myosin bundle around the periphery of the cell with nascent and very dynamic lamellipodia trying to go off and protrude. But then what happens is those lamellipodia are consumed by this contraction. You literally see how actin filaments that are trying to grow, they are just collapsed into the bundle all the time. And so we thought, based on our work on keratocyte, that if we strengthen myosin or weaken adhesion, then this cell will self-polarize. However, the shocking result was that instead of increasing myosin strength, in fact, myosin inhibition leads to polarization and motility initiation. And the shapes of stationary cell and moving cells are actually very keratocyte-like. So instead of we used drugs that strengthen myosin, nothing happened. And in control also, the cell never lost polarity. But if you inhibit myosin, for example, with blebbistatin, then the cell starts moving. Okay, so obviously the mechanism is somehow different. So the hint to what this mechanism is, one of the hints is that if you look at actin, for example, in the process of polarization, when you add this blebbistatin inhibiting myosin, so before this inhibition, the region near the leading edge where the actin-myosin bundle is very rich with actin, and there's less actin at the region where dynamic lamellipodia are, and less actin in the middle of the cell. But then actin disappears from the bundles, and before appearing at the in the new massive lamellipodium, it increases in the middle of the cell. So kind of hinting that there are two networks at the leading edge, this nascent lamellipodia and actin-myosin contractile bundles, and they compete for the same pool of actin. So hinting that there's some kind of competition here between two networks. And so from all these clues, we put together a model which qualitatively is very simple. So there are two types of actin networks at the periphery, branched and bundle, and they compete. So competition is, there are two main competitive parts other than competing for common actin. One is branched filaments are collapsed into bundles simply by action of the myosin. And there's also the branched filaments are fighting back in a very subtle way. So if branching filaments starting to get dense and thin, they start protruding this part of the boundary, leaving the bundles behind. So in a way, they blow the bundles away from the leading edge, creating the centripetal flow. An important thing about it is that movement of the boundary of the cell affects this competition. So imagine that the cell moves this way. So first it's shown with a dashed contour and then with solid contour. At the rear, this movement benefits the bundles because this movement would compress all the branched actin here into the bundles. At the front, movement helps the branched actin because it leaves the bundles behind, but growing branched actin filament can stay here. And then actin density determine how the boundary moves, because if you have a lot of branched filaments, they would be moving the boundary, but bundles tends to contract and bring in the rear. And so you probably already can figure out how the model works, but just in case, I'll show you the equations. Equations look kind of messy, but in fact, if you look at them, those of you who did math biology for a while will immediately recognize that the main thing here is a model from ecology. It's competition for resource model between two species. You can just imagine that one network is one type of animal, another type of animal, and they compete. So there's terms like that which describe this competition. And so not many people saw how this model, so this model, of course, depending on the parameter, can predict either coexistence of two networks or one network winning. Okay. And here, unfortunately, in this model, we don't know really the parameters. We have this freedom to choose whatever parameters we want. But what is important is the following. Now we solve this model on a contour on the boundary of the cell, and we have effective diffusion because branched filaments are growing slightly sideways and kind of propagate to the sides, and in the bundles, actin reshuffles the actin filaments. And if you put this model in space and add diffusion to it, then there are actually some relatively kind of obscure theorems. I don't think many people know about them, but the theorem is basically like that. You cannot make one network win at the left of the domain and another at the right of the domain. There's just no way. This model always such that if the model without diffusion, without space, predict that one network wins, then it has to win on the whole domain. Okay. However, in our model, what changes this fact is that the coefficients describing the competition of this network don't have to be constant everywhere, because when the cell starts moving, right, then one network is advantaged at the front, another at the rear, and so that's the answer. Then qualitatively it's clear that we can find the parameters such that when the cell starts moving, then branch network will win at the front, bundled network will win at the rear, and here you are. And so indeed, when we did this on the moving boundary, it turned out to be the case. So that's the prediction. So far it's rather qualitative predictions, which probably could be figured out without modeling. However, the model makes two rather non-trivial predictions. So one is what happens when we start varying myosin strength. And so in the model, what myosin does, the higher the myosin, the more bundles are advantaged. And so when we looked at this, it turns out that the polarization is predicted for either for intermediate strength of myosin. For very high myosin and very low myosin, there's no polarization. And why is that? Because at very high myosin, bundles win everywhere, no matter what. For very low myosin, protrusion wins everywhere, no matter what. So when we tested this using variable concentration of blibistatin, indeed this is the case. For intermediate myosin, cells move nicely. For low myosin, cells do not polarize. For very high myosin, they are not disc-like, of course, but they're not really moving. They just become this ugly creatures creating protrusions everywhere and not able to move coherently. Another very non-trivial prediction is that in keratocyte, if you think about it, in keratocyte, to polarize, you don't need the cell to move. So in a way, first you create this mechanical polarization inside, and then the cell starts moving. Here, it turns out you need movement to polarize. So the prediction is if you have somehow immobilize the cell, there will not be cytoskeletal polarization inside. And indeed, that's the case. So the experiment was to put the cell into fabricated microwell of the size roughly of the cell. So the cell cannot move, it bumps into the walls. And indeed, in this cell, when blibistatin was used, there was no cytoskeletal polarization. So here, movement is an integral part of the self-polarization. And so that shows that even in such simple cases, the mechanisms of polarizations are completely different for rather similar type of epithelial cells, just showing that how hard our life will be when we'll go to really complicated cells in 3D. So that's pretty much it. Here are some lessons from all these projects that I showed you, and that's pretty much it. So those are people who did all this work. These people helped us with free boundary simulations. Galvanotaxis experiment were done by my postdocs at UC Davis. This guy did modeling for the first and third project, and experiment for the first project with keratocyte was done by this group at Stanford, and last project for rat liver cell by this group at Harvard. Thank you.
Nice talk.
Thanks.
So in your models, in the images that you show from the cells, normally you see some elongated attachments opposite from the leading edge, maybe some surface attachments, and I was wondering if how those might be affecting the membrane tension and how those might affect your results, your model.
What do you mean elongated attachments?
So if the cell is moving to the right, normally on the left you see a very thin and long membrane attachment that clips eventually.
You're talking about keratocyte or the
You
last
see
cell?
those in both cells?
Both? So you meant
Yes, for example,
did you mean these guys?
Yes.
Oh no, so those are there's some micro adhesions left at the surface, and those are kind of membrane tubes that are left behind. Sometimes people call them retraction fibers.
Mm-hmm.
There's some evidence that they're absolutely irrelevant. They're not interesting. Real adhesions are close to the edge of the cell, around the edge, right here. So separately, I have somewhere images of actually, I have images of adhesions.
But you'd imagine that.
So here's the adhesive structures.
Mm-hmm.
Those are sometime there's a whole zoo of different types of adhesions, and in some cells there are so-called focal adhesions, and it's those that when you pull hard you strengthen them. In keratocytes, the adhesions are not focal adhesions. They are so-called nascent adhesions, kind of less mature than focal adhesions. So
I was just wondering if those affected the membrane tension term that you had in your modeling.
no. So what we do for membrane adhesion, membrane tension, we just assume that it's a certain function of area of the cell. But that's a valid question. How do you find membrane tension is a very tricky thing. So if actin is binding to the membrane, that has to be taken into account. There is some indirect evidence that in keratocytes there's very little attachments between actin and membrane, so we just take in-plane membrane tension. But it's a valid concern for more complicated cells.
So in the first part, you're talking about the spontaneous polarization, and in the model, you basically gave a little bump to make the myosin start to move.
Mm.
But I may have missed what's the bump in real life?
Yeah, that's a great question. We actually, like the movie that I showed. This is without any initial bump. What we did here, we included some noise, some artificial noise into transport of myosin initially. So it effectively creates some fluctuation here, fluctuation there, and eventually the cell moves. What we found from the simulations that the stationary symmetric state is locally stable. If you keep this fluctuation small, it will always stay stationary. But if you go over the top, and over the top doesn't mean really the amplitude, you rather need to create some aggregates of myosin which are big enough and long-lasting enough. Then the cell starts deforming. And so the great question is what's happening in real life? What are these fluctuations? Clearly, lots of things are fluctuating in the cell, but so far we don't know. We don't know what creates those fluctuations. They're definitely not the small number fluctuations. If there were, for example, just 100 adhesive molecules, then clearly just Poisson fluctuations would create some condition. but number of important molecules are in thousands and tens of thousands, so it's easy to estimate that they will not create this inhomogeneities. Something else is in play. Sure.
So when you're actually looking at it in real life,
Mhm.
how long does it take for you to for it to start?
So yeah, as I showed, spontaneous polarization takes of the order of 30 minutes, like 30 minutes, 40 minutes, and that's it's meaningful number. When we change condition, for example, strength in myosin, then this time goes down, weakened myosin, this time goes up. So there's something important about this time.
So I have two questions. The first maybe a short answer. So in the final part of your talk, does the bundled actin ring, is it physically coupled to the membrane by say some protein attachments or?
Probably not.
Okay.
So like from high resolution microscopy, we kind of see membrane flapping around.
Okay,
Yeah,
so
it's certainly adhering to the surface
Okay.
through the ventral membrane. But,
Okay.
yeah.
Okay, so that was the first question. The second question is more general.
Mhm.
So how do you think about now transitioning to 3 dimensions, which you've alluded to, and what aspects of your models do you expect
Yeah,
to translate?
great and painful question. Painful because there are more and more really high-profile cell biologists who start doing working on three-dimensional migration, saying that there are absolutely no lessons from 2D, just none. Which I don't think is true, but well, people are entitled to their opinions. So the only thing we did so far, those collaborators of ours on the second project, they looked how this rat liver cell polarizes in 3D. So they put it in this engineered scaffold, and the result is the same as in 2D. By itself, the cell never polarizes. If you weaken myosin, it polarizes and starts moving. And the mechanism we're pretty much sure is the same as we predicted. There is this also competition between two types of the networks. And so one of the things which I have no doubt survives in 3D is is this very simple paradigm that when the cell moves, myosin is swept to the rear, and it creates contraction at the rear, pulling the rear forward. What probably doesn't survive in 3D, it's not this actin filament growth that pushes the leading edge forward, but rather hydrostatic pressure created by this contracting myosin at the rear that pushes the cell forward. But this is not for sure. So yeah. From what I know, there are already some good data, good experiments on migration of the cell in 3D. There's pretty much nothing known about polarization in 3D.
Following up the 3D question, so it is known nowadays that cells migrate differently. If we're talking about tumor cell migration, you have genetic modified cells are genetic different from migration differentiates from localization on the tumor.
Right.
So how can you consider that in your model, thinking about the myosin, since there's
Right.
a lot of this in the field right now?
So from the data I see in the literature, one thing which is pretty constant for all this 3D migration, either whether those cancer cells or something else, is that myosin is always a must. Like the very simple experiment, inhibit myosin, any cell in 3D stops. While in 2D, when we inhibit myosin, the cell goes like nothing happens. And so what I didn't mention is what the mechanism I told you about is just a couple of redundant mechanisms that cannot polarize the cell but can keep it moving. In 3D, myosin is a must. No myosin contraction, the cell doesn't move. But there's of course a big difference between, for example, cancer cells that migrate through dense tissue and some other types of 3D migration. So there are about 6 to 10 hypothesized mechanical mechanisms how the cell moves. But of course, on top of this mechanics, there's this mind-boggling complexity of regulating, of biochemical regulation of those mechanical modules. And so when you combine those 6-10 mechanisms with close to 100 signaling pathways. There's enormous variety. And so that, I don't know if that's the answer, but that's kind of the beginning of big conversation.
Actually, that's a nice segue to my question here, because in your model for the keratocyte, you implied that the viscosity of the actin filament is constant throughout the cell and just depends on the flow of the actin. But it's well known that the actin, the structure of the actin network changes, and you mentioned this yourself, that near the lamellipodium you have a lot more dense actin than further back. And so how, so while your model nicely explains this, is it really realistic for a cell that it's moving because the actin organization changes so dramatically?
Yeah. So this is an easy question. As I mentioned, this model is minimal, and indeed viscosity, for example, depends on density of actin. So we have more sophisticated models when we have equation for actin density, and their viscosity is function of density. And those models qualitatively predict the same thing. So I just didn't go into that because this is a relatively secondary effect to the main effect. So it works for whatever viscosity.
Well, except for this one example where you had the pimentin that you need to include
Vinculin.
to explain the behavior of the cell, because you need to capture
Right.
the specific behavior of the actin in certain places.
Right. So what more extensive work shows that mechanical properties of adhesions are essential. Viscosity is not very essential. So it's not adhesion is not really actin property. It's like interaction of actin with other type of system. What we don't know, of course, how this adhesion properties depend on actin density. That's kind of unknown. We didn't include it into the model. But so basically the answer is that mechanical properties of actin network are, I mean, that's what the model can say. Mechanical properties of actin network are not essential. The model works whatever they are. We can make it viscoelastic, we can increase viscosity, decrease viscosity, it still works. Make viscosity dependent on density, make it non-uniform, still works the same. But properties of adhesion make it very are essential. They change behavior qualitatively. And experimentally, of course, it's a tough thing to test if this is actually true. And of course, another huge simplification is actin network is not anisotropic. It's not isotropic, really. And this anisotropy, we have one of the models when we investigated anisotropy, and it doesn't seem to be important for polarization, but we're not completely sure. There are many types of fancy anisotropy that could affect it.